Hello Bruins, and today we're going to talk about cell respiration. And before we can talk about cell respiration, we need to have an idea about how life follows the laws of thermodynamics and how we convert energy into cellular work, and what role the ATP plays, and the three key phases of cell respiration. And we'll just end talking about glycolysis. All right, now when we think about energy being used in living things, we think about food chain. And food chain is a good place to start because that's how energy flows through the ecosystem, where you have here the producers, and the producers again convert that light energy by photosynthesis into food. And then the food chain is basically the flow of feeding, succession of feeding of organisms. So producers are fed, feeds. The next level, which we call trophic levels or eating levels, is our primary consumers. And our primary consumers feed on these producers. And then these primary consumers are then consumed by our next feeding level, which we call secondary consumers. I'm sure I write it out, secondary. And then our secondary consumers are, if they're fed by our tertiary consumers, and further, if they are fed upon our quaternary consumers. So as you can see, as we go up this levels, trophic levels, the amount of energy that started at producers decreases, and as you probably remember, probably the rule of 10. So just as everything else in the universe follows these key laws of thermodynamics, the first law is, if you can remember, the energy cannot be destroyed or created, but energy can be transformed, or and whenever it is transformed, some of the energy is always lost as heat. And that adds to the disorder to the universe. So as you can see, the ultimate source of energy in life is the sun, right? And that provides the energy for our producers to convert carbon dioxide and water with light energy to make food. And that food or chemical energy is then used by our consumers. Also, other terms that you should remember is producers are also called autotrophs, and consumers are called heterotrophs. Okay. Now, energy available to do work is sometimes also referred as free energy. So, and also you could think of like potential energy. So here, this girl is on the top of the slide. She has the ability to move. She has more free energy than if she was on the bottom of the slide. Also, in terms of energy, when you have high concentration of solutes in one end and you have low here, you have a disequilibrium. And because of this equilibrium, you have the potential to do work. Versus here, when things have reached equilibrium, there's less potential, or less free energy. Same thing in terms of chemical. Here you have a compound that has a lot of bonds, and these bonds represents energy. And when these bonds are broken down to simpler molecules, it has less free energy, or less ability to do work. Now, if you think of the free energy or the ability to do work is also in terms of disequilibrium. So when you have disequilibrium, you can harness that to do work. So for example, here you have a turbine that's run by water, right? So here, if you have water, you have more water here and less water here, that turns a turbine. But then what happens when you reach equilibrium? It can no longer do work. So life is more where you have to constantly put energy in, right? In terms of here it's water, and as you keep putting water, then this turbine keeps running, right? So what's the difference between this and this picture? So here, the turbine, again, you have this constant flow of energy, but this energy is being released in multi-steps, versus this is in one big step. So in terms of life, life is more like this multi-step open hydroelectric system. Because if you think about it, you don't want to release energy all at once. You want to release energy all in small steps. Now, as living organisms, we get our energy from chemical energy, and that's from our foods. So if you look at these molecules, if you remember from our biochemistry unit, this is our fat molecule, and notice there's a lot of these carbohydrate bonds. And then you have your starch here, or in other words, your polysaccharide. And here we have our monosaccharide. Now, which molecule would have the greatest amount of energy? And if you look at in terms of energy is in our bonds, you'd probably say the fat molecule because it has a lot of this, and you'd be correct. So the lipids have the most stored energy, say, versus monosaccharide and polysaccharide. And then if you compare these two, of course, polysaccharide has more energy compared to our monosaccharide. And always think that this energy is again in our bonds. It's stored in these bonds. So energy flows through the ecosystems, and two key process that's involved is photosynthesis, which takes that light energy, converts it to food by using carbon dioxide and water. And then you have glucose and oxygen produced from photosynthesis, which are used in the process of cellular respiration to break that energy that's in this glucose molecule to release ATP. And ATP is a key energy currency in living things. And of course, whenever you're converting energy, you always lose some energy as heat. Now let's talk about ATP. And ATP is a nucleic acid. As you can see, there's a ribose sugar, a nitrogen base called adenine, and 3 phosphate groups. And where the energy is stored are in these bonds between the phosphate groups. And notice the negative bonds. So these negative bonds, negative charges repel each other. So there's a lot of strain in these bonds. So when this bond is broken, a lot of energy is released. So how is energy released? As you see, you add water by the process of hydrolysis. You can break that phosphate bond, release an inorganic phosphate, and then you have ADP and energy. And then that's how energy is released. Now, if you want to store back energy into ATP by condensation, and energy input, we put that energy back into ATP. Now, how does ATP do cellular work? What happens is energy is very exergonic, right? So when you if you remember, like in transport, when you phosphorylate this protein, it has a conformational change. And when you break that bond, it releases energy, and you have another conformational change, and it transports the bond. Same thing also in terms of motor proteins. By phosphorylating the motor proteins, it changes its shape, and then by dephosphorylating, it changes its shape again, and thus you have movement. Same thing here with chemical. And if we wanted to combine X and Y, that's a very endergonic reaction, meaning you got to input a lot of energy. But by phosphorylating it, we make it highly energized, unstable, reactive. And when that bond breaks, it releases energy, and that energy is used to make that bond. So, to understand cellular respiration, let's first talk about mitochondria and the different parts of the mitochondria. So if you remember in class, we use an example of taking a bag and putting another bag in another bag, so a double bag. So think of a mitochondria as a double bag, where the outer bag is your outer membrane, and then your second bag is your inner membrane. And then inside the second bag is the matrix, and the space between the two bags are the intermembrane space. And also these folds of the inner membrane is called cristae. So where does cellular respiration take place? Well, there's 3 phases. The first phase is called glycolysis, and basically glycolysis takes that glucose molecule, and breaks it down to a 3-carbon molecule called pyruvate. So it's taking that 6-carbon molecule and breaking it down to a 3-carbon molecule. And it produces a little bit of ATP and a high energy carrier called NADH, which will later go to the electron transport chain. And the next phase is called the Krebs cycle, where the glucose is completely broken down, releasing the carbons as CO2 and make a little bit of ATP, but a whole bunch of these high electron carriers, NADH and FADH, which then go on to the electron transport chain, which is the third phase. And in the third phase is where you make the most ATP, where the last electron acceptor is your oxygen, and when that accepts those electrons, it becomes water. Now this whole process cannot occur without oxygen. So here, per glucose molecule, you make about 2 ATPs for glycolysis, and about 2 ATPs from Krebs cycle, and about 32 ATPs from the electron transport chain. Now, we talked about this high electron carrier, NAD+, and basically it'll accept these electrons and it becomes NADH. So think of gaining electrons is like gaining energy. Now you also hear this word oxidize and reduce. The word oxidize means losing electrons. And when you gain electrons, it's referred as reduce. So you might see these two words, which you'll probably learn in chemistry. And this means gaining electrons. Also, you could think of, again, our electron carriers as a taxi. When it picks up the electrons, it's loaded, and then it drops off the electrons. So our two main high electron carriers is our NAD and FAD. Okay. So NAD, when it picks up those electrons, becomes NADH, a highly energized molecule. And FAD is another electron carrier, and when it picks up electrons, it becomes FADH2. Alright, so now let's talk about the first phase of cellular respiration, which is glycolysis. And glycolysis is basically taking that 6-carbon molecule glucose, and in order to start the reaction, you need to input energy. And you input energy with 2 ATPs. Alright, and then you break that sugar into 2 pyruvate acid, and at the end you produce 4 ATPs plus 2 NADHs. So basically you put in 4 ATPs and you get back 2 ATPs. So we get a net gain. So basically we're saying we get a net gain of 2 ATPs and 2 NADH. Now, glycolysis is, again, does not need oxygen, and if there is no oxygen, it'll go into what's called fermentation or anaerobic respiration. In anaerobic respiration, basically what you want to do is further continue glycolysis. Now, in order for glycolysis to happen, you need NAD+. So here you have 3-carbon molecule pyruvic acid and NADH, and you release one of those carbons as carbon dioxide, and you result with a 2-carbon molecule and NAD+. And this happens with yeast to make beer or bacteria to make wine. You also have a different type of lactic acid, like in animals or different bacteria. Here, our 3-carbon pyruvic acid becomes lactic acid, where using electron from the NADH to recycle NAD+. So here's another way of looking at fermentation, or in other words, anaerobic respiration. And remember, anaerobic respiration means no oxygen. Okay, and it's happening in the cytoplasm. So, and remember, glycolysis happens in the cytoplasm. So again, with glycolysis, you produce some ATP, and you make a 3-carbon molecule called pyruvate. Now, in order to keep glycolysis going, we need NAD+. So in alcoholic fermentation, you remove that carbon as carbon dioxide, you end up with a 2-carbon intermediate, and then again, using another electron, you recycle NAD+ to make a 2-carbon alcohol called ethanol. Oops. Okay, so let's summarize glycolysis. In glycolysis, you need glucose, a 6-carbon molecule, and you make some ATP. And if you remember, we input 2 ATPs. At the end, we get 4 ATPs with a net gain